Okay, so now that we talked about proteins and the different structures from our primary structure to our quaternary structure, we're going to talk a little bit about how that relates into the actual function. And so myoglobin and hemoglobin are important molecules in the body, and they have different quaternary structures, and they have very different functions in the body. So again, to reiterate, the quaternary structure, I'll just write 4. The quaternary structure is going to be interactions between multiple polypeptides. And remember, each of these polypeptides started as a primary structure sequence, and each of these polypeptides folded into their secondary structure, their alpha helixes and their beta sheets. And then each of these secondary structures went on to fold further due to the different amino acid side chains and their interactions through those van der Waals forces, hydrogen bonding, dipole-dipole interactions. And then multiple of these tertiary structures are now getting together to interact and form the quaternary structure. So a hemoglobin is a tetramer. And a tetramer just means that it has 4 regions. And with the case of hemoglobin, it's going to have 2 alpha chains, and it's going to have 2 beta chains. And then you have myoglobin, and this is going to be a globular protein. And this is going to have 1 polypeptide chain. And it's going to have 1 oxygen binding site. All right, so here's myoglobin. It has its proximal histidine that's going to help with the ability to bind oxygen. And then here's hemoglobin, and it has 4 binding sites. The 4th one is hidden, 2 betas and 2 alpha chains. Now we have hemoglobinopathies in 2 different types. We have qualitative and quantitative. So of course, qualitative means its shape, its folding, how well can it bind to oxygen, etc. And sickle cell anemia is an example of a qualitative. You change the shape of the hemoglobin. So now it can't bind oxygen as well, but it has other properties that provide protection, such as provide protection from malaria. And then quantitative is how much is being made. So the quantitative side of things is going to be called a thalassemia. So just as a recap, remember that we have our hyperbolic myoglobin. It gets saturated very fast. And then we have our sigmoidal hemoglobin, where it has cooperativity. That first binding to oxygen takes a lot longer. And then once that first oxygen binds, it sort of goes over this initial hurdle, and the others can bind. And so this tetramer inhibits binding of oxygen at low partial pressure of oxygen. So tissues have low partial pressure. So you don't want your hemoglobin to bind oxygen in tissues. Your lungs have a higher partial pressure of oxygen. So, oops, I should put this up, sorry. This is where the partial pressure of oxygen. So lungs has a much higher, your tissues have a much lower, and so your tissues will not in your tissues, your hemoglobin will not bind oxygen, but in your lungs, your hemoglobin will bind oxygen. So this is a typical type of allosteric protein. And this means you have alteration in protein activity through binding of an effector molecule. Now, the tetrameric structure of hemoglobin is going to be, again, that one that facilitates its ability to saturate with oxygen in the lungs, and then its ability to release this oxygen in the capillaries where there's low partial pressure oxygen. So when the pressure of oxygen is high in the lungs, hemoglobin is saturated. When the pressure of oxygen in the tissue is low, hemoglobin can't bind as well. Myoglobin, however, can bind well, and myoglobin is going to be high in our muscle tissues, such as your heart. And when the muscle will bind, or the myoglobin will bind to the oxygen released by the hemoglobin, and it'll kind of store that oxygen in muscle tissues until it's needed. So for your oxygen binding, you have myoglobin's tertiary structure has 8 alpha helixes that are connected by coils, and this is referred to as the globulin fold. So it has no beta sheets in the myoglobin. The helixes are going to create hydrophobic oxygen binding pockets within the heme center. The heme is what we call a planar porphyrin ring. And it has a pyrrole ring with nitrogen in the center. Okay, so it has negative charge groups are going to interact with histidine and arginine. And then you also have hydrophobic regions of methyl and vinyl in heme. And all in all, you have a total of 16 interactions with your myoglobin. So I didn't even see that I had this already written here. Just chatting away. We'll bring the heme down to heme section. So myoglobin again has no beta sheets, and then the heme has the porphyrin ring with the pyrrole rings with the nitrogen in the center. And your negative charges are going to be interacting with histidine and arginine, which are in the pocket. And then you have about 16 interactions within this molecule that make oxygen bind. Okay, so in general, oxygen is going to bind to your iron directly, and this is going to be on in the planar ring, the planar porphyrin ring. And so you're going to have your oxygen bind. Oops. And then this is going to lead to a chelation of 6 ligands. You have 4 nitrogens, 1 oxygen, and histidine. And this is also a nitrogen of histidine. And then when histidine binds to the iron, it pulls the iron above the plane, and then oxygen binding will bring iron back into the plane. And this is going to occur in both myoglobin and hemoglobin. And so the geometry of the molecule will change depending on histidine binding and then the presence of oxygen. And then so this is going to cause a conformational change. And in myoglobin, this conformational change associated with histidine, iron, and oxygen has no effect on myoglobin. So myoglobin has no effect. And then so that is why our myoglobin is going to have a hyperbolic curve. For hemoglobin, you have to break a salt bridge. And so this is why it's sigmoidal. There is a hurdle to reach in order for that oxygen to bind. Now there is cooperative binding of oxygen and hemoglobin. And so this is where you are going to get different states of the molecule. So when no oxygen is bound, it is said to be at a tense or taut state. And that's this state up here. And then as you change from your tense state to your R state, which is a relaxed state, your hemoglobin has a higher affinity for oxygen. So breaking that salt bridge requires energy. And so therefore, the first O2 is slow. The second O2 through the 4O2 are faster because you're already at the relaxed state. So now everything kind of just opens up. And so this is called positive cooperativity, and this leads to the sigmoidal oxygen saturation curve. Now another function, form and function can be described with antibodies. Antibodies are immunoglobulins. They bind to antigens. They're part of the immune response. They have 2 heavy and 2 light chains. The heavy chains are the ones that go all the way through the arms to the trunk. So you have your arms and the base is the trunk. And then your light chains are the ones that are only part of the arms. So that's why they're called light, they're smaller. You're going to have constant domains and variable domains. So you see here we have our variable light, variable heavy, variable light, variable heavy. And then we have our constant light. And we have our constant heavy. All right, and so there are more constant regions in your heavy chain than in your light chain, but the variable regions are the same. Now, oops, wrong green. It's a heavy chain. Okay, so it's your variable region that is going to be your antigen binding site, shown here. So for example, if we had here's our heavy, here's our light. At the end, we'll have this square, and it's going to be the same on both sides. So if you had antigens that were square, they could bind. If you had antigens that were circles, they cannot bind into this square, but they might have another antibody that has circle-shaped. And then now your circles can bind to the circle-shaped antibody. And so the specificity of the binding is on those variable regions, and the variable regions made up of both the heavy and the light chain. Now the light chain and heavy chain are going to be made up of different domains. And as the immunoglobulin folds, you're going to have a beta barrel made up of numerous beta sheets. So light plus heavy have domains, and they're going to have beta barrel, which has many beta sheets. Okay, and then we have our chaperones. Chaperones are also called heat shock proteins. And they're going to be important because they help other proteins fold. And they're often going to be induced after stress. One famous one is heat shock protein 70, and this keeps nascent proteins from folding. And nascent means new, newly made. Heat shock protein 60 helps proteins refold. And this uses ATP. So where would you see this? So as an example, you have your mitochondria genes, and they come from the nucleus. And so our mitochondria genes, we'll put them as red. They will be grabbed by heat shock protein 70. They will be brought to the mitochondria. So here's our mitochondria. And then in the mitochondria, you will get your protein. You will have your, do a different color, heat shock protein 60. And this will refold that protein. So what this makes sure, sometimes this is important because you have genes that are going to be made by your nucleus, but they actually need to function somewhere else. So these heat shock protein 70 will keep that protein from folding until it gets to where it needs to go. And once it gets where it needs to go, another set of heat shock proteins will help that protein fold and then become functional. Now, denaturing of proteins is where you have native protein structures and you're altering them. And this is important because denaturing changes the biologic activity. However, what's equally important, your primary structure is not disrupted. So we went from primary, which is a sequence, to our secondary, to our tertiary, to our quaternary. Well, denaturing will break down all of these, but your primary is intact. So that's why when you're add heat or acidic or basic, your proteins fall apart except for that primary structure is pretty stable. And so it does not usually get disrupted. And I'd say, and I'll put usually. I mean, of course, when you push it too far, obviously you're going to break peptide bonds. So following denaturization, proteins can return. So when you return to normal, your proteins can refold. But under extreme conditions, again, it's going to become irreversible. So here's some denaturing elements. We can have heat. Heat are going to break those hydrogen bonds. And remember, hydrogen bonds are going to be not only important for the side chain, so in that tertiary, it's also important for the structure of the secondary. So you're going to break down your tertiary, which obviously will break down your quaternary, and then your secondary will break down, but not your primary. UV is going to have a similar as heat, ultraviolet radiation. Strong acids or bases, you're going to have disruption of hydrogen bonds, salt formation, etc. Urea, again, going back to hydrogen bonds. Organic solvents are going to change dielectric constants, hydration of ionic groups. So again, those side groups in those tertiary structures are going to be affected. And agitation, again, gets back to hydrogen bonds. So some non-enzymatic modifications can change functions of proteins as well, and some of them are on purpose. We can have glycosylation, adding sugars, oxidation, and these can lead to a functional loss. Sometimes you add things to control proteins. So you make the protein, but you don't want it to work too quickly, so you glycosylate it. There's going to be slow turnover of proteins. So we have collagen or hemoglobin. These are slow turnovers, which means they last a relatively long time. So these are going to be, oops, that's a horrible thing, sorry, glycosylated. So they don't get broken down, they don't get used up too quickly. And then this, of course, glycosylation is going to depend on our sugars. So sugar levels in the blood can bind to proteins groups as well. So if you have hyperglycemia, this is going to result in an increase of glycosylation. And so not only are you going to have your hemoglobin and collagen glycosylated, but you're going to have proteins that aren't supposed to be glycosylated, have sugars added to them. Now you also have temperature, of course, pH, solvents can denature proteins as well. And then finally, let's just talk very briefly about prions. Prions are misfolded, called infectious proteins, and they cannot be degraded. These proteins are dominant. They cause normal prion proteins to misfold. So prion proteins are in our brain, they're in everybody's brain, and they function through, they're thought to be important in the synapse, so the sending of signals, neurologic signals. And so you have your prion protein, which is a misfolded protein. So this is a prion protein SC. SC is for scurvy. So let's see, PRPSC equals, and scurvy is a disease of sheep. So that's where it was discovered. And so this scurvy protein is going to be dominant over the normal protein, the control protein, and it's going to cause that normal protein to misfold. The misfolded protein has more beta sheets. And so it completely changes the secondary structure, which changes the tertiary structure, and this leads to aggregates. You can also have genetic mutations that can increase chances of misfolding of proteins as well. Some neurodegenerative diseases associated with prion proteins, one of the famous is mad cow disease. We have scurvy that I just talked about, or screepies. We have elk disease, which is associated with wasting, so skinny, poor little elk, and Creutzfeldt-Jakob disease for humans.